Meiosis is the cell division of gametes, which results in four haploid daughter cells. You receive a copy from your father and one from your mother. And synapsis is the connecting of homologous chromosomes. And crossing over is the sharing of genetic material. Meiosis 1 then results in recombinant chromosomes. In meiosis 2, we have four haploid daughter cells, two of which are recombinant. Hemophilia is an X-linked genetic disorder. Hemophilia is a disorder in which your blood doesn't clot normally because it lacks sufficient blood clotting proteins. For instance, if the mother has hemophilia and the father does not, if you're a girl, you would receive a normal X copy from your father and an affected X chromosome from your mother, and you would be a carrier for hemophilia. If you're a boy, you would receive a Y copy from your father and an affected X chromosome from your mother, and you would have hemophilia. You receive a copy from your father, one from your mother. The DNA then replicates, and crossing over occurs, and then you have your recombinated chromosomes. You have your recombinated chromosomes, which leads to genetic variation. Recombination is the rearrangement of genetic material, especially by crossing over in chromosomes or by the artificial joining of segments of DNA from different organisms. In hemophilia, it would be the rearrangement of the chromosomes you receive from your parents. Ready? Here we have the mother, who is a carrier for hemophilia, and a father who is normal, he doesn't have hemophilia. They can either have a normal son or a son who is affected by hemophilia. Hemophilia arises from a mutation in the FA gene that affects the clotting factor proteins. Because of this, people with hemophilia don't have clotting proteins and they bleed out. It's an X-linked genetic disorder that mostly affects males. Females are usually carriers of the gene, and in rare cases, they have hemophilia. Symptoms of hemophilia includes double vision, neck pain, and prolonged headaches, blood in the stool and urine, faintly bleeding, tender muscles and bruises, swollen, painful joints, bleeding of the gums, tongue, or mouth, and in some severe cases, bleeding in the brain can cause long-term problems such as seizures and paralysis. This disorder can also lead to death if the bleeding cannot be stopped or if it occurs in vital organs such as the brain. Treatment for hemophilia includes demand therapy and replacement therapy. In replacement therapy, clotting factors V3 or clotting factors 1X are injected into the vein. This form of treatment is preventative and provides the body with clotting agents that will prevent you from bleeding out. And demand therapy is used in case of an emergency. For example, you cut your finger and then you inject clotting factors 1X or V3. Those at risk are individuals who have hemophilia.
Mitosis is cell division that results in two daughter cells. It grows and replaces worn-out cells. Mitosis is controlled by genes that are found inside every cell, and this control can sometimes go wrong, leading into mutations. Normal mitosis results in cell division. And sometimes when a cell is wrong, it goes through apoptosis. But in cancer, it starts replicating really fast without stopping. Synovial sarcoma is a rare cancer which occurs near the joints of the arms, neck, or leg. The tumor grows near the tissues around these joints, and that's basically where the cell replication occurs. There's a genetic mutation in the cells that cause it to replicate without stopping. Synovial sarcomas display characteristics of chromosomal TX18 translocations that result in enhanced transcription of the insulin-like growth factor 2 IGF-2 gene. IGF-2 is an essential fetal mutagen involved in the pathogenesis of different tumors, leading to cellular proliferation and inhibition of apoptosis. Under normal physiological conditions, the balance between the expression and activities of the proteins involved in the pathway is tightly controlled. The IGF system has been implicated in various pathophysiological conditions and is thought to play an especially prominent role in tumorigenesis. Density inhibition is when cells stop dividing once they form a layer. Cancer cells do not obey these rules, and they continue to divide, and they start to pile up. And this is what occurs in synovial sarcoma, usually in the tissues near the joints, and what forms a large mass of tumor that continues to grow. Rather than responding to the signals that cause normal cells to cease proliferation and enter the G0 phase, tumor cells generally continue growing to high cell densities in culture, mimicking their uncontrolled proliferation. As I said, in synovial sarcoma, this occurs in the tissues around the joints of the arms, neck, or legs. Therefore, a large mass is formed. Anchorage dependence is when cells anchor to a dish surface and divide. Cancer cells do not exhibit anchorage dependence. They don't cling to each other, and it makes it easier to metastasize to different areas of the body. This happens about 50% of the time in synovial sarcoma. Although evidence in the literature strongly suggests a primary role of SYTSX in the induction of synovial sarcoma, the pathway from SYTSX expression to cellular transformation is unknown. Several studies involving microarray analysis on synovial sarcoma tumors and cell lines have identified several potential downstream targets of the SYTSX. There are no well-established risk factors for synovial sarcoma, but the disease is associated with a chromosomal translocation TX18, P11, Q11. This means that parts of chromosomes 18 and chromosome X have switched places in synovial sarcoma tumor cells. It is not known whether this mutation occurs randomly or follows a specific chain of events. Because of this translocation, synovial sarcoma cells contain a mutant gene. This mutant gene is thought to contribute to the development of the disease. Synovial sarcoma is a malignant tumor of the tissues around the joints. A malignant tumor is cancerous and can metastasize, which means that it can multiply uncontrollably and move to various parts of the body. This tumor can often metastasize into the lungs. A benign tumor is non-cancerous and lacks the ability to metastasize and can be removed easily through surgery. But synovial sarcoma is a malignant tumor that can metastasize. Synovial sarcoma can be treated with surgery, radiotherapy, and chemotherapy. Radiotherapy is usually used in order to reduce the risk of leaving cancerous cells behind. Chemotherapy might be recommended, but it's still unknown if it works by preventing metastasis. Prognosis in synovial sarcoma patients is influenced by the quality of surgery patients receive and the characteristics of the disease. Patients with small tumors can be completely removed with adequate margins at the diagnosis have an excellent prognosis. The risk of developing distant metastasis is higher for patients with tumors that are larger than 5 centimeters.